Okay, so welcome back to this next video in which we are discussing Pavlovian, if you like, classical conditioning. Right, okay, so we have now discussed action potentials and the release of neurotransmitter from an axon terminal in response to an action potential arriving in that axon terminal. Okay, what we now want to see is how serotonin acting on receptors on the axon terminal is going to increase the amount of neurotransmitter that axon terminal releases. Okay, right. So basically, on the surface of the axon terminal, you're going to have receptors to serotonin. So let's just draw the setup again. So here is the axon terminal of our L1 sensory neuron. Okay, then we have our axon terminal of our L29 neuron. Okay, so this is this interneuron which becomes activated when we shock the head of the Aplysia californica. Okay, and basically the L29 neuron is going to release serotonin onto the axon terminal of the L1 sensory neuron. So firstly, I would just like to discuss the structure of serotonin with you. Then we'll discuss the pathway which it's going to induce within the L1 sensory neuron axon terminal. Okay, so serotonin is also called 5-hydroxytryptamine. And basically, it's based on the structure of the amino acid tryptophan. Okay, so before we can discuss the structure of 5-hydroxytryptamine, which is also usually just abbreviated to 5H for hydroxy rather, and then T for tryptamine, we need to discuss the structure of the Pyro ring and also the Indole ring. Okay, so let's discuss the structure of the Pyro ring, basically. So, the Pyro ring basically is an aromatic ring containing 4 carbons and then a single nitrogen. Okay, so here it is. It's a 5-membered ring. We have these 4 carbons here and then a nitrogen. Okay, and I'm drawing the skeletal structure, so you don't show carbons. Carbons are implicitly shown by the corners of the ring. Okay, and you don't show hydrogens coming off carbons either. So where carbons have missing bonds, those are implicitly to hydrogens, but you show everything else. So we'll have a hydrogen coming off this nitrogen like so. So we have a double bond here and a double bond here, so it's beautifully symmetric. So this is a Pyro ring. Now we're going to take it up a step. We're going to go on to the structure of an Indole ring. Okay, so basically an Indole ring is a Pyro ring with a benzene ring attached to it. So let me show you the structure of a Pyro ring. So here is a Pyro ring, like so. We have a double bond there, with a hydrogen coming off it. And then we have this benzene ring coming off the side of the Indole ring here. So here is this 6-membered benzene ring, and we'll have alternating double and single bonds. Okay, so there's a double bond here, remember. So this is the structure of an Indole ring. Now what we're going to see is that tryptophan contains the Indole ring. Okay, so if we now show the structure of tryptophan. Okay, right. So we'll start with the core amino acid structure, and then the R group of tryptophan is basically going to be an Indole ring. So here's the amino group, like so. Then we've got the alpha carbon of the amino acid here, with a hydrogen coming off it. Then we've got the carboxylic acid group down here. Okay, and the R group of tryptophan consists of a methylene group, and then with this Indole ring coming off the side, I'm suddenly going to revert back to drawing skeletal structures rather than molecular structures. Okay, so here we go. Here is my skeletal structure. So here's my Pyro ring. Okay, so we're connected to this carbon up here of the Indole ring. Okay, so here's my double bond, here's a double bond, there's the hydrogen coming off the nitrogen, and then we need a benzene ring coming off the side here. So here's this 6-membered ring, double bond there, this is the Indole ring. So this is tryptophan. Now, to convert this into tryptamine, what you have to do is take off the carboxylic acid group that comes off the alpha carbon here and replace it with a hydrogen. But we don't want just tryptamine, we want 5-hydroxytryptamine. Now, to understand what 5-hydroxytryptamine is, we need to know the numbering system of the members of this indole ring. So, if you were going to name the elements within this indole ring, how would you do it? Well, we've got 1, 2, 3, 4, 5, 6, 7, 8 carbons. So which one stands out? It's the nitrogen. So we're going to start with the nitrogen. We'll call that number 1. Then we just go round in order. We go 2, 3. Now this is slightly confusing. When you've got carbons which are in 2 rings, you don't call this carbon 4, you call it 3A. Okay, this next one then is called 4. This one up here is called 5. This one's 6, 7, and again this one, you don't call it 8, but instead 7A. Okay, so that's the numbering system for tryptophan. Now what we know, that's the numbering system for the members of the indole ring. Now we know exactly what 5-hydroxytryptamine is going to be. We need to take the carboxylic acid group off and replace it by a hydrogen, and then we need to stick an alcohol group coming off the 5th carbon of the indole ring. Okay, so here is the amino group. Okay, here's the alpha carbon, which will have 2 hydrogens coming off, here and here. So we've replaced the carboxylic acid group with a hydrogen. Okay, then we have a methylene group here, and then we have the indole ring coming off here. Okay, so this 5-membered ring with a nitrogen at the head here, and a hydrogen coming off it. We have a double bond there, a double bond there. Okay, then we have the benzene ring here. We have a double bond there, and then we have an alcohol group coming off this 5th carbon here. So this is 5-hydroxytryptamine. 5-hydroxytryptamine. 5-hydroxytryptophan would be where we still have the carboxylic acid group here. Okay, so 5-hydroxytryptamine then, or serotonin. So this is what the L29 axon terminal is releasing onto the axon terminal of the L1 sensory neuron. Okay, right. So the L1 sensory neuron has a receptor for serotonin, which is a G protein coupled receptor. Okay, so it's got these 7 characteristic membrane spanning alpha helices. So this is a 5-hydroxytryptamine receptor. Okay, right. So let me just get a bit more paper and then we'll continue the discussion of this. So, basically, the serotonin is going to come in and bind to the G protein coupled receptor, which has these 7 membrane spanning alpha helices, like so. And here's the carboxylic acid group of the G protein coupled receptor, and here's the amino group. So in comes serotonin, and it will bind to the serotonin receptor on the surface of the axon terminal of the L1 sensory neuron, and it will trigger a conformational change in this G protein coupled receptor that then allows it to interact with a heterotrimeric G protein. Okay, so remember, heterotrimeric G proteins consist of 3 subunits, and they're pretty much the same in Aplysia as they are in humans. Okay, so we have an alpha, a beta, and a gamma subunit like so. And basically the alpha subunit can be in 2 states. It can either be in the on state, in which it will have GTP bound to it, or it can be in the off state, in which it has GDP, guanosine diphosphate, bound to it. And when the alpha subunit is in the off state and has guanosine diphosphate bound to it, then it will associate with the beta-gamma complex here. So the beta-gamma complex is this name for a beta subunit bound to a gamma subunit. And the beta and gamma subunit never ever break apart. Okay, so they're often considered together as the beta-gamma complex. So basically what will happen is when serotonin binds to the G protein coupled receptor here, which is the serotonin receptor on the surface of this axon terminal, then the G protein coupled receptor will change conformation, and it will now be able to bind to the heterotrimeric G protein here. And what it will do is it will catalyze the release of the guanosine diphosphate by the alpha subunit and the binding of a guanosine triphosphate molecule, GTP, from the cytoplasm. so that you get the alpha subunit bound to GTP. That turns the alpha subunit into the on state, basically. And when the alpha subunit is in the on state, it no longer wants to associate with the beta gamma subunit. So the two will separate off. So you'll get this alpha subunit with GTP bound and a beta gamma subunit, which will go off. This alpha GTP subunit is then going to go and activate adenylyl cyclase enzymes. Okay, right, so the next topic for us is adenylyl cyclase enzymes within Aplysia. Okay, right, so basically what we know is that there are 4 adenylyl cyclase enzymes within Aplysia. Okay, and these are called adenylyl cyclase AC, and then you put Aplysia, APL, and then there's adenylyl cyclase Aplysia 8. There is adenylyl cyclase Aplysia A, adenylyl cyclase Aplysia B, then there is adenylyl cyclase Aplysia C, and then finally adenylyl cyclase Aplysia D. So there are 4 adenylyl cyclases which have been found within Aplysia. Contrast that to the 9 which are known within humans. Okay, now what's nice is that from what we know, they seem to have very similar structures to the human adenylyl cyclases. Okay, so I'll now show you the structure of a human adenylyl cyclase, but we think that the Aplysia adenylyl cyclases are the same. Okay, so you have the amino terminus on the intracellular aspect, and I should just say adenylyl cyclase enzymes are integral membrane proteins. They sit within the cell membrane. Okay, so here's the amino terminus on the intracellular aspect of the membrane. Then you have a cluster of 6 membrane-spanning alpha helices, then a special loop, and then another cluster of 6 membrane-spanning alpha helices, and then the carboxylic acid terminus down here. So this is the structure of a human adenylyl cyclase enzyme, but we think, and we have good evidence in favor of these adenylyl cyclase enzymes having a very similar structure to this. Basically, if you look at the primary sequence of amino acids within the polypeptides for these, you can basically see that the residues conform to the structure that you would expect if they have this sort of membrane-spanning topology. Now, what do I mean by that? Well, basically, if you are going to be a residue within a membrane-spanning alpha helix, you generally have to be hydrophobic, because the cell membrane consists of a huge number of hydrophobic molecules. So if you're going to sit in there comfortably, you generally have to be hydrophobic. So what we can do is we can look at the primary sequence of these proteins, and that's not too difficult to do. Okay, so you can look at the primary sequence of amino acids within the polypeptide, and what you can say is, are there clusters of amino acids which are all hydrophobic and then could be a membrane-spanning alpha helix? So for instance, if you found clusters of hydrophobic residues like so, okay, and I'll complete it up now, so we need 12 of them. So there we go, I've drawn 12 of these little orange things. If you find 12 clusters of hydrophobic residues along the polypeptide length, then you can say, well, this is probably the first membrane-spanning alpha helix here, this is probably the second membrane-spanning alpha helix here, this is probably the third one, etc. And you can start to get some idea maybe of the membrane-spanning topology of these things. Basically, when they did this for these adenylyl cyclases, which they found within the Aplysia californica, they found that it did indeed have 12 regions which looked like membrane-spanning alpha helices, which is very hopeful for suggesting that actually these things have a very similar structure to the human adenylyl cyclases. Okay, so let me just talk about the human adenylyl cyclase more, and you should be aware that although Aplysia californica is highly studied, it's not as highly studied as Homo sapiens. Okay, so we don't know as much about the adenylyl cyclases in Aplysia californica as we do about the human ones. Okay, right, so basically, this cluster of 6 membrane-spanning alpha helices in a human adenylyl cyclase enzyme is called transmembrane domain 1, TMD1. This cluster of 6 membrane-spanning alpha helices over here is called transmembrane domain 2, TMD2. Now, although TMD1 and TMD2 look mightily impressive, they are not the actual active adenylyl cyclase enzyme. The active adenylyl cyclase enzyme is in these cytoplasmic portions. These transmembrane domains just latch the whole thing at the cell membrane. Okay, so basically, this loop between transmembrane domain 1 and transmembrane domain 2 is known as the C1 domain. Okay, so this is the C1 domain. And this carboxylic acid tail over here, so the C-terminal tail here, is known as the C2 domain. Okay, now C1 and C2 are further divided up. Okay, C1 is divided up into C1a and C1b, while C2 is further divided up into C2a and C2b. Okay, now the active adenylyl cyclase enzyme is made by the dimerization of C1a with C2a. So basically, the structure I've drawn is an inactive adenylyl cyclase. To turn this adenylyl cyclase on, you have to dimerize the C1a with the C2a domains. You have to bring them close together, join them together, and that will make you a functional enzyme which will start catalyzing the cyclase reaction. Okay, so this is all known in humans. We can speculate that it's the same in these adenylyl cyclases in Aplysia. Okay, and there is evidence, as I say, there's good evidence that it does seem to be that way. Okay, so what will happen then to turn it into the active form is that we'll need to get the dimerization of C1a with the C2a domain. Okay, so let me show this here. Now I've used a little bit of artistic license here. I have hugely stretched C2a from what it was in this previous picture to show the two stuck together. Here's C1a, here's C2a. Okay, and they're now dimerized together. And this active form of the adenylyl cyclase will then start catalyzing the cyclase reaction. Now, what is the cyclase reaction? Well, basically, you take in adenosine triphosphate, ATP, and you give out cyclic AMP plus pyrophosphate, which is often abbreviated to PPI. Now, let's have a look at this reaction in a bit more detail. So, adenosine triphosphate, often abbreviated to ATP. Let's have a look at the structure of this. So basically, in order to understand the structure of adenosine triphosphate, we need to know what adenosine is. Okay, so basically adenosine means adenine bound to a ribose sugar. Okay, so the organic base adenine, which I'm just going to demote to being represented as a green rectangle. Okay, bound to a ribose sugar, that is what is known as adenosine. Okay. So, the blue pentagon with the 5th carbon sticking off up here, this is supposed to represent ribose, okay, and the green rectangle with an A written in it, that's supposed to represent, green triangle did I just say, green rectangle with an A written in is supposed to represent adenine. So, the combination of the two is then adenosine. So we now want adenosine triphosphate, so we need to attach 3 phosphate groups onto the ribose sugar to turn it into adenosine triphosphate. Okay, so I will colour in those 3 phosphate groups in bright pink here. So, how do you then convert adenosine triphosphate into cyclic AMP and pyrophosphate? What is the structure of cyclic AMP and pyrophosphate? So basically, a pyrophosphate molecule is just 2 phosphate groups linked together. So the first, well, actually, let me just tell you some nomenclature concerning these phosphate groups on adenosine triphosphate. So the first phosphate group of adenosine triphosphate is called the alpha phosphate. The second phosphate is called the beta phosphate, and the third phosphate is called the gamma phosphate. Okay, right. So to turn adenosine triphosphate into cyclic adenosine monophosphate and pyrophosphate, what you do is you cut the bond between the beta phosphate and the alpha phosphate, and therefore you end up with 2 phosphate groups linked together, the beta phosphate and the gamma phosphate. Okay, and this molecule of two phosphate groups linked together is called pyrophosphate and is often abbreviated to PPI. Meanwhile, cyclic adenosine monophosphate, often abbreviated to CAMP or called cyclic AMP, this basically is not just what is left. Okay, so if you just leave adenine bound to ribose bound to a single phosphate, that would just be adenosine monophosphate. We need to create a new cycle in this molecule to turn it into cyclic adenosine monophosphate. So we need to bind this alpha phosphate to this third carbon's alcohol group here to create an extra cycle. So let me show you the structure of this. Okay, right, so here's the ribose sugar, here's the fifth carbon, and now the phosphate group coming off the fifth carbon is going to be bound to that third carbon down there to create an extra cycle within this structure basically, and that is now called cyclic adenosine monophosphate. Okay, so there's the only phosphate group. Then here we have the ribose sugar here. Then we have the adenine organic base in green here. Okay, so that's the reaction that adenylyl cyclase enzymes catalyze. So, in humans, what happens is you have the alpha S subunit, which then comes and binds to both the C1 domain and the C2 domain, holds them close together, and then promotes the dimerization of the C1a and the C2a domains to produce the active enzyme. This alpha subunit within Aplysia is going to do a very similar thing to the human G-alpha S subunit. Okay, it's going to promote the dimerization of C1a and C2a, and then promote the formation of the active enzyme, so the active enzyme will then start churning out cyclic AMP. Now what does cyclic AMP do? Well, it's going to go on and activate protein kinase A. So let's just discuss protein kinase A. And again, we'll discuss protein kinase A in humans, but we think the Aplysia form is very, very similar. Okay, so in humans, protein kinase A consists of two regulatory subunits and two catalytic subunits. Okay, so basically protein kinase A is often called an R2-C2 complex because it consists of two regulatory subunits, that's the R2, and then two catalytic subunits, that's the C2. So these two structures that I'm drawing here, these are the two regulatory subunits. So this is a regulatory subunit and this is a regulatory subunit. So I'll write their names out in full. This is a regulatory subunit of protein kinase A, of pKa. Okay, and I'm going to colour them in as well. We'll have them coloured in red. Okay, so in red here, these are the regulatory subunits of protein kinase A. Now these little invaginations that I've left here, these little holes down here, these are meant to represent the cyclic AMP binding sites. So you'll see that each regulatory subunit of protein kinase A has two cyclic AMP binding sites. So basically, two cyclic AMP molecules can bind in here, and two cyclic AMP molecules can bind, one in here and one in here. And that will trigger conformational changes in the regulatory subunits, and we'll come back to that in a moment. Now, at the moment, we are looking at the inactive protein kinase A enzyme. Okay, so this is inactive protein kinase A. And basically, the regulatory subunits sequester another subunit called the catalytic subunit of protein kinase A, which I'll show here in green. Okay, so in green, these are the catalytic subunits of protein kinase A. So, in the inactive form, protein kinase A assembles one of these R2-C2 complexes. So you have these two regulatory subunits bound together, and then each of them has a catalytic subunit bound to them. Now, the regulatory subunits are not involved in the phosphorylation of proteins. It's the catalytic subunits which can go off and phosphorylate serine and threonine residues within proteins. Okay, so these are the actual kinase portions. However, when the catalytic subunits are bound to the regulatory subunits like so, they are not active. They can't actually phosphorylate serine and threonine residues. However, when cyclic AMP goes up, what will happen is cyclic AMP will bind in these cyclic AMP binding sites here. So this is a cyclic AMP binding site, and basically that will trigger a conformational change in the regulatory subunit of the protein kinase A. Okay, so let me show this very vividly for you. So I'll have it going from being in this sort of L shape to being linear. Okay, like so. And when the regulatory subunits undergo this conformational change, they let go of the catalytic subunits, and the catalytic subunits can then go off and phosphorylate serine and threonine residues. And each one of these catalytic subunits is an enzyme of its own accord. Okay, so each one of them goes off on their own and then can phosphorylate serine and threonine residues. So you don't need two of them to be able to do it. Okay, right. So these little balls that I'm showing here that I'm now colouring in orange, those are meant to now represent the cyclic AMP molecules. So cyclic AMP has been demoted somewhat from being represented by this to being represented just as an orange blob. Okay, so basically the regulatory subunits will change conformation when cyclic AMP binds, and this will release the catalytic subunits, and the catalytic subunits will then go and phosphorylate serine and threonine residues. Now, one of the things that the catalytic subunits are going to phosphorylate is a special potassium channel, and this is going to be how activation of this 5-hydroxytryptamine receptor is going to actually lead to an increase in the release of neurotransmitter from the axon terminals of these L1 sensory neurons. Okay, so the catalytic subunit of protein kinase A is going to phosphorylate a special channel within the axon terminal of our L1 sensory neuron. Okay, and this is a special type of potassium channel. Okay, so here it is. I'll colour it in pink. So in pink here, this potassium channel is called the S channel. Okay, so this is called the S channel. And basically, the S stands for serotonin sensitive. So this is the serotonin sensitive potassium channel. Serotonin sensitive. Okay, right. And basically this serotonin sensitive potassium channel is normally open within the cell membrane of the axon terminal. Okay, so it's one of these leaky channels, it's always open. However, once protein kinase A catalytic subunit phosphorylates this S channel, this serotonin sensitive potassium channel, okay, it will cause the channel to close. Okay, and this is what's going to cause an increase in the release of neurotransmitter. The reason being that it is going to cause the prolonging of the action potential within the axon terminal. Okay, and that's going to lead to increased activation of the voltage-gated calcium channels. Okay, so let me explain this further. Right, so you will remember that in order to couple the action potential to the release of synaptic vesicles, okay, so let me just draw this picture again. So here's our axon terminal. In order to get the action potential to trigger the release of synaptic vesicles, what has to happen is the action potential has to propagate into the active zone, as it's called. So the active zone is just the name for this portion over here, the presynaptic membrane with the docked synaptic vesicles. Okay, and there what will happen is the membrane undergoing an action potential will activate voltage-gated calcium channels, and these voltage-gated calcium channels will open and calcium will come in, and the amount of calcium you let in determines how many synaptic vesicles you're going to release. Okay, so basically closing this S channel is going to cause the action potential to prolong, which means that the time period for which the electrical potential difference across the cell membrane is depolarized is going to increase. So the voltage-gated calcium channels, which are only active when the membrane is depolarized, either only open when the membrane is depolarized, they will remain open for longer now that we've prolonged the action potential. Okay, so you'll let in more calcium, the The more calcium you release, the more synaptic vesicles you release, hence the more neurotransmitter you release. So I want to now explain to you why closing this S channel is going to lead to prolonging of the action potential. So here's our diagram of the action potential back again. So remember, in this repolarizing portion of the action potential, we opened voltage-gated potassium channels, and these allow potassium to move out. Okay? Now, the S channel is not a voltage-gated potassium channel. It's a leaky one. It's always open. But do you think it's not conducting potassium as well in this repolarizing phase? Okay, it's playing its part. We focus on the voltage-gated potassium channels because they're the ones which are changing. Okay, they're the ones which only become open in this repolarizing portion of the action potential. But actually, this S channel will be open through the entire action potential. And when the electrical potential difference is at, for instance, plus 40 millivolts up here, or in fact is anywhere along this level here, you will be getting potassium moving out of the S channel, just as you get potassium moving out of the voltage-gated potassium channels. And this movement of potassium out through the S channel will contribute to the repolarizing phase of the action potential. So, if you now close this S channel, then the amount of potassium that will be moving out of the cell during the repolarizing phase will be reduced. Okay, so the rate at which potassium is moving out of the cell in the repolarizing phase will be reduced, and that means that it will take longer to repolarize the membrane. So maybe the action potential will now look something like this instead. Okay, so once you've closed that S channel, it goes to this thing in turquoise here, so it prolongs basically. The action potential now lasts longer. So closing these S channels is going to cause the prolonging of the action potential because of the contribution of these S channels to the repolarization. Okay, so prolonging of action potential. Now, if the action potential is prolonged, then the time for which you will be beyond threshold potential for the voltage-gated calcium channels, which will generally be around the same number that we've always been working with. So negative 40 millivolts is the threshold potential for the activation of voltage-gated sodium channels. We saw that it was also the threshold potential for the activation of voltage-gated potassium channels. And generally, it's also the threshold potential for the activation of voltage-gated calcium channels. And you can see that in this prolonged action potential, the time for which you are beyond that threshold potential will be greater than in the shorter action potential, where it stops here. Okay, so those voltage-gated calcium channels will be open for this entire period, and therefore they'll be open for longer. So the amount of calcium they will let in is going to be greater, and therefore you'll release more neurotransmitter. So overall, this is the molecular mechanism by which sensitization occurs of the Gill withdrawal reflex. Basically, the release of the serotonin by the L29 interneuron onto the axon terminal of the L1 sensory neuron is going to lead to the prolonging of the action potentials within the axon terminal, which will lead to a greater amount of calcium coming in through the voltage-gated calcium channels, which will then lead to a greater amount of exocytosis of neurotransmitter because you'll exocytose a greater number of synaptic vesicles. Okay, so just to recite the bigger picture again, that means now that when you stimulate the L1 sensory neuron by putting your water onto the siphon, it will release a greater amount of neurotransmitter onto the L7 motor neuron, which means the L7 motor neuron will fire a higher frequency of action potentials, which means that the gill will now contract more. And that's the basis of how the gill withdrawal reflex is sensitized by shocking the head of the Aplysia californica. In the next video, we'll now discuss Pavlovian conditioning.